Cell cycle control system. Network of regulatory proteins that governs progression of a eukaryotic cell through the cell cycle. Cell division. Separation of a cell into two daughter cells. In eukaryotic cells, it entails division of the nucleus, mitosis, closely followed by division of the cytoplasm, cytokinesis. Cell junction. Specialized region of connection between two cells or between a cell and the extracellular matrix. Cell line. Population of cells of plant or animal origin capable of dividing indefinitely in culture. Cell locomotion. Active movement of a cell from one location to another. Cell memory. The ability of cells and their descendants, without undergoing any change of DNA sequence, to retain a trace of the effects of past influences, displaying the consequences in persistently altered patterns of gene expression. Cell signaling. The molecular mechanisms by which cells detect and respond to external stimuli and send messages to other cells. Cellulose. Structural polysaccharide consisting of long chains of covalently linked glucose units. It provides tensile strength in plant cell walls. Cell wall. Mechanically strong fibrous layer deposited by a cell outside its plasma membrane. Prominent in most plants, bacteria, algae, and fungi, but not present in most animal cells. Central dogma. The principle that genetic information flows from DNA to RNA to protein. Centriole. Short cylindrical array of microtubules, usually found in pairs at the center of a centrosome in animal cells, also found at the base of cilia and flagella, and called basal bodies. Centromere. Constricted region of a mitotic chromosome that holds sister chromatids together, also the site on the DNA where the kinetochore forms and then captures microtubules from the mitotic spindle. Centrosome. Cell center. Centrally located organelle of animal cells that is a primary microtubule organizing center and separates to form the two spindle poles during mitosis. In most animal cells it contains a pair of centrioles. Centrosome cycle. Duplication of the centrosome during interphase and separation of the two new centrosomes at the beginning of mitosis to form the poles of the mitotic spindle. Channel. An aqueous pore in a lipid membrane with walls made of protein, through which selected ions or molecules can pass. Checkpoint. Point in the eukaryotic cell division cycle where progress through the cycle can be halted until conditions are suitable for the cell to proceed to the next stage. Chemical bond. Chemical affinity between two atoms that holds them together. Types found in living cells include ionic bonds, covalent bonds, and hydrogen bonds. Chemical group. Set of covalently linked atoms, such as hydroxyl group, OH, or an amino group, NH2, that occurs in many different molecules and the chemical behavior of which is well characterized. Chemiosmotic coupling. Mechanism in which a gradient of hydrogen ions, a pH gradient across a membrane, is used to drive an energy-requiring process, such as ATP production or the transport of a molecule across a membrane. Chiasma, plural chiasmata. X-shaped connection visible between paired homologous chromosomes in division 1 of meiosis, and which represents a site of crossing over. Chlorophyll, light-absorbing pigment that plays a central part in photosynthesis. Chloroplast, specialized organelle in algae and plants that contains chlorophyll and in which photosynthesis takes place. Cholesterol, lipid molecule with a characteristic four-ringed steroid structure that is an important component of the plasma membranes of animal cells. See Figure 11.7. Chromatid, see sister chromatid. Chromatin, complex of DNA, histones, and non-histone proteins found in the nucleus of a eukaryotic cell, the material of which chromosomes are made. Chromatin remodeling complex. Enzyme, typically multi-subunit, that uses the energy of ATP hydrolysis to alter histone DNA interactions in eukaryotic chromosomes. The resulting alteration changes the accessibility of the underlying DNA to other proteins, including those involved in transcription. Chromatography. General term for a type of technique used to separate molecules in a mixture on the basis of their size, charge, or their ability to bind to a particular chemical group. In a common form of the technique, the mixture is run through a column filled with a particular matrix that is designed to bind or let through the desired molecule. Chromosome. Long, thread-like structure composed of DNA and associated proteins that carries the genetic information of an organism, especially visible when plant and animal cells undergo mitosis or meiosis. Chromosome condensation. Process by which a chromosome becomes packed into a more compact structure prior to M phase of the cell cycle. Citric acid cycle, TCA, or tricarboxylic acid cycle, Krebs cycle. Central metabolic pathway in all aerobic organisms that oxidizes acetyl groups derived from food molecules to CO2. In eukaryotic cells, these reactions are located in the mitochondrial matrix. Cilium, plural cilia, hair-like extensions on the surface of a cell with a core bundle of microtubules and capable of performing repeated beating movements. Cilia, in large numbers, drive the movement of fluid over epithelial sheets, as in the lungs. Cis. On the same side, for example, the cis-Golgi network is that part closest to the endoplasmic reticulum. Clathrin. Protein that makes up the coat of one type of transport vesicle. Clathrin-coated vesicles bud from the Golgi apparatus on the outward secretory pathway and bud from the plasma membrane on the inward endocytic pathway. Cloning. Making many identical copies of a cell or a DNA molecule or an organism. Coated vesicle. Small membrane-enclosed organelle with a cage of proteins, the coat, on its cytosolic surface. It is formed by the pinching of a protein-coated region of membrane. Codon. Sequence of 3 nucleotides in a DNA or messenger RNA molecule that represents the instruction for incorporation of a specific amino acid into a growing polypeptide chain. Coenzyme A, CoA. Small molecule used in the enzymatic transfer of acyl groups in the cell. See also acetyl-CoA and Figure 336. Cohesin. Protein complex that forms a ring that holds sister chromatids together after DNA has been replicated in the cell cycle. Coiled coil. Especially stable rod-like protein structure formed by 2 or more alpha helices coiled around each other. Collagen. Fibrous protein rich in glycine and proline that is a major component of the extracellular matrix and connective tissues. Exists in many forms. Type 1, the most common, is found in skin, tendon, and bone. Type 2 is found in cartilage. Type 4 is present in basal laminae, and so on. Combinatorial control. Describes the way in which groups of proteins work together in combination to control the expression of a single gene. Complementary. Describes 2 molecular surfaces that fit together closely and form non-covalent bonds with each other. Examples include complementary base pairs, such as A and T, and the 2 complementary strands of a DNA molecule. Complementary DNA, cDNA. DNA molecule made as a copy of mRNA and therefore lacking the introns that are present in genomic DNA, used to determine the amino acid sequence of a protein by DNA sequencing or to make the protein in large quantities by cloning followed by expression. Complex. A collection of macromolecules that are bound to each other by non-covalent bonds to form a large macromolecular structure. Associations of proteins are called protein complexes. Associations of protein and nucleic acids are called nucleoprotein complexes. Complex trait, a heritable characteristic whose transmission to progeny does not obey Mendel's laws. Such traits, for example height, usually result from the interaction of multiple genes, each of which is inherited in Mendelian fashion. Condensation. See chromosome condensation. Condensation reaction. Type of chemical reaction in which two organic molecules become linked to each other by a covalent bond with non-committant removal of a molecule of water. Condensin. Protein complexes with a ring-like structure that help carry out chromosome condensation. Conformation. Spatial location of the atoms of a molecule relative to each other. The precise shape of a protein or other macromolecule in 3 dimensions. Connective tissue. Tissues such as bone, tendons, and the dermis of the skin, in which extracellular matrix is plentiful and carries the mechanical load. Conserved synteny. Regions of the genome where corresponding genes are in the same order in the species being compared. Coupled reaction. One of a linked pair of chemical reactions in which free energy released by one reaction serves to drive the other reaction. Coupled transporter. Member transport protein that carries out transport in which the transfer of one molecule depends on the simultaneous or sequential transfer of a second molecule. Covalent bond. Stable chemical link between two atoms produced by sharing one or more pairs of electrons. Crossing over. Process whereby two homologous chromosomes break at corresponding sites and rejoin to produce two recombined chromosomes, resulting in a physical exchange of DNA segments. Cyclic AMP, CAMP. Nucleotide generated from ATP in response to hormonal stimulation of cell surface receptors. CAMP acts as a signaling molecule by activating protein kinase A. It is hydrolyzed to AMP by a phosphodiesterase. Cyclic AMP-dependent protein kinase, protein kinase A, pKa. Enzyme that phosphorylates target proteins in response to a rise in intracellular cyclic AMP concentration. Cyclic photophosphorylation. Photosynthetic process involving photosystem 1 only, in which chloroplasts can generate ATP without making NADPH. Cyclin. Protein that periodically rises and falls in concentration in step with the eukaryotic cell cycle. Cyclins activate specific protein kinases, see cyclin-dependent protein kinases, and thereby help control progression from one stage of the cell cycle to the next. Cyclin-dependent protein kinase, CDK. Protein kinase that has to be complexed with a cyclin protein in order to act. Different CDK cyclin complexes trigger different steps in the cell division cycle by phosphorylating specific target proteins. Cytochrome. Colored, heme-containing protein that transfers electrons during cellular respiration and photosynthesis. Cytokine. Small protein made and secreted by cells that acts on neighboring cells to alter their behavior. Cytokines act via cell surface cytokine receptors. Cytokinesis. Division of the cytoplasm of a plant or animal cell into two, as distinct from the division of its nucleus, which is mitosis. Cytoplasm. Contents of a cell that are contained within its plasma membrane, but, in the case of eukaryotic cells, outside the nucleus. Cytoskeleton. System of protein filaments in the cytoplasm of a eukaryotic cell that gives the cell shape and the capacity for directed movement. Its most abundant components are actin filaments, microtubules, and intermediate filaments. Cytosol. Contents of the main compartment of the cytoplasm, excluding membrane-enclosed organelles such as endoplasmic reticulum and mitochondria. The cell fraction remaining after membranes, cytoskeletal components, and other organelles have been removed. DAG. See diacylglycerol. Dalton. Unit of molecular mass, defined as 1/12 the mass of an atom of carbon-12, 1.66 times 10 to the negative 24 grams, approximately equal to the mass of a hydrogen atom. Denature, to cause a dramatic change in conformation of a protein or nucleic acid by heating it or by exposing it to chemicals, usually results in the loss of biological function. Deoxyribonucleic acid, cDNA. Desmosome, specialized cell-cell junction, usually formed between two epithelial cells, characterized by dense plaques of protein into which intermediate filaments in the two adjoining cells are inserted. Detergent, soapy substance used by biochemists to solubilize membrane proteins. Diacylglycerol, DAG, lipid produced by the cleavage of membrane inositol phospholipids in response to extracellular signals, composed of two fatty acid chains linked to glycerol. It serves as a membrane-located signaling molecule to help activate protein kinase C. Dideoxy-DNA sequencing, the standard method of DNA sequencing. It utilizes DNA polymerases and chain-terminating nucleotides. Differentiation, process by which a cell undergoes a progressive change to a more specialized and usually easily recognized cell type. Diffusion, the spread of molecules and small particles from one location to another by random, thermally driven movements. Dimer, a structure composed of two halves. A homodimer is composed of two identical subunits, a heterodimer of two different subunits. Diploid, a cell or organism containing two sets of homologous chromosomes and hence two copies of each gene or genetic locus. See also haploid. Disulfide bond, SS bond, covalent linkage formed between two sulfhydryl groups on cysteines. Common way to join two proteins or to link together different parts of the same protein in the extracellular space. Divergence, the differences due to mutation that accumulate in two DNA sequences derived from a common ancestral sequence. DNA, deoxyribonucleic acid, double-stranded polynucleotide formed from two separate chains of covalently linked deoxyribonucleotide units. It serves as the cell's store of genetic information that is transmitted from generation to generation. DNA cloning, see cloning. DNA library, collection of cloned DNA molecules representing either an entire genome, genomic library, or copies of the mRNA produced by a cell, see DNA library. DNA ligase, see ligase. DNA methylation, the enzymatic addition of methyl groups to cytosine bases in DNA. Methylation generally turns off genes by attracting proteins that block gene expression. DNA microarray, a glass slide upon which a large number of short DNA molecules, typically in the tens of thousands, have been immobilized in an orderly pattern. Each of these DNA fragments acts as a probe for a specific gene, allowing the RNA products of thousands of genes to be monitored at the same time. DNA polymerase, see polymerase. DNA repair, collective term for the enzymatic processes that correct deleterious changes affecting the continuity or sequence of a DNA molecule. DNA replication, the process by which a copy of a DNA molecule is made. DNA transcription, see transcription. Domain, small discrete region of a structure. A protein domain is a compact and stable folded region of polypeptide. A membrane domain is a region of bilayer with a characteristic lipid and protein composition. Double bond, a type of chemical linkage between two atoms formed by sharing 4 electrons. Double helix, the typical conformation of a DNA molecule in which two polynucleotide strands are wound around each other with base pairing between the strands. Dynamic instability, The property shown by microtubules of growing and shrinking repeatedly through the addition and loss of tubulin subunits from their exposed ends. Dynein. Member of a family of large motor proteins that undergo ATP-dependent movement along microtubules. Dynein is responsible for the bending of cilia. Electrochemical gradient. Driving force that causes an ion to move across a membrane, caused by differences in ion concentration and its electrical charge on either side of the membrane. Electron. Fundamental subatomic particle with a unit negative charge, E-. Electron acceptor. Atom or molecule that takes up electrons readily, thereby gaining an electron and becoming reduced. Electron carrier. Molecule such as cytochrome C that transfers an electron from a donor molecule to an acceptor molecule. Electron donor. Molecule that easily gives up an electron, becoming oxidized in the process. Electron transport chain. A series of membrane-embedded electron carrier molecules along which electrons move from a higher to a lower energy level, as in oxidative phosphorylation and photosynthesis. Electrophoresis. Technique for separating mixture of proteins of DNA fragments by size and electric charge, by placing them on a polymer gel and subjecting them to an electric field. The molecules migrate through the gel at different speeds depending on their size and net charge. Electrostatic attraction. Attractive force that occurs between oppositely charged atoms. Examples are ionic bonds and the attractions between molecules containing polar covalent bonds. Element. Substance that cannot be broken down to any other chemical form, composed of a single type of atom. Embryonic stem cell. ES cell. An undifferentiated cell type derived from the inner cell mass of an early mammalian embryo. Embryonic stem cells can be maintained indefinitely as a proliferating cell population, cell line in culture, but remain capable of differentiating, when placed in an appropriate environment, to give any of the specialized cell types in the adult body. Endocytosis. Uptake of material into a cell by an invagination of the plasma membrane and its internalization in a membrane-bounded vesicle. See also pinocytosis and phagocytosis. Endoplasmic reticulum, ER. Labyrinthine, membrane-enclosed compartments in the cytoplasm of eukaryotic cells, where lipids and secreted and membrane-bound proteins are made. ES cell. See embryonic stem cell. Endosome. Membrane-enclosed compartment of a eukaryotic cell through which endocytosed materials passes on its way to lysosomes. Enhancer. Regulatory DNA sequence to which transcription regulators bind, influencing the rate of transcription of a structural gene that can be many thousands of base pairs away. Entropy. Thermodynamic quantity that measures the degree of disorder in a system. The higher the entropy, the more the disorder. Enzyme. A protein that catalyzes a specific chemical reaction. Epigenetic inheritance. Inherited that is superimposed on the information inherited in the DNA sequence itself. Often, information in the form of a particular type of chromatin structure, e.g., a certain pattern of histone modification or DNA methylation. Enzyme-coupled receptor. Transmembrane receptor proteins that activate an intracellular enzyme, either a separate enzyme or part of the receptor itself, in response to ligand binding to the extracellular part of the receptor. Epithelium, plural epithelia. Sheet of cells covering an external surface or lining an internal body cavity. Equilibrium. In a chemical context, a state in which two or more reactions are proceeding at such a rate that they exactly balance each other and no net chemical change is occurring. Equilibrium constant, K. A number that characterizes the steady state reached by a reversible chemical reaction, given by the ratio of forward and reverse rate constants of a reaction. See Table 3.1, page 96. ER. See endoplasmic reticulum. Escherichia coli, E. coli. Rod-like bacterium normally found in the colon of humans and other mammals and widely used in biomedical research. Eubacteria, the proper term for the bacteria of common occurrence, used to distinguish them from archaea. Eukaryote, living organism composed of one or more cells with a distinct nucleus and cytoplasm. Includes all forms of life except archaea and bacteria, the prokaryotes, and viruses. Euchromatin, one of the two main states in which chromatin exists within an interphase cell, the other being heterochromatin, characterized by particular histone modifications and associated proteins. Genes in euchromatin are in general able to be expressed. Evolution, the gradual change in living organisms taking place over generations that results in new species being formed. Exocytosis, process by which most molecules are secreted from a eukaryotic cell. These molecules are packaged in membrane-enclosed vesicles that fuse with the plasma membrane, releasing their contents to the outside. Exon, segment of a eukaryotic gene that is transcribed into RNA and expressed. Dictates the amino acid sequence of part of a protein. Exon shuffling, evolutionary process by which new genes form by linking together combinations of initially separate exons encoding different protein domains. Extracellular matrix, complex network of polysaccharides such as glycosaminoglycans or cellulose and proteins such as collagen secreted by cells. A structural component of tissues that also influences their development and physiology. Extracellular signal molecule, any molecule present outside the cell that can elicit a response inside the cell when the molecule binds to a receptor protein. Some signal molecules, such as steroid hormones, can enter cells and act on internal receptors, whereas others, such as proteins, act as receptors embedded in the plasma membrane and exposed on the cell surface. FAD, see FADH2. FADH2, reduced flavin adenine dinucleotide, a major electron carrier in metabolism produced by reduction of FAD during the oxidation of catabolites such as succinate. Fat, lipids used by living cells to store metabolic energy, mainly composed of triacylglycerols. See panel 2.4, pages 70 to 71. Fatty acid, compounds such as palmitic acid that has a carboxylic acid attached to a long hydrocarbon chain, used as a major source of energy during metabolism and as a starting point for the synthesis of phospholipids. See panel 2.4, pages 70 to 71. Feedback inhibition, a form of metabolic control in which the end product of a chain of enzymatic reactions reduces the activity of an enzyme early in the pathway. Fermentation, the breakdown of organic molecules without the involvement of molecular oxygen. This form of oxidation is less complete than in aerobic processes and yields less energy. Fertilization, sequence of events that starts when a sperm cell makes contact with an egg and leads to their fusion and further development. Fibroblast, common cell type found in connective tissue that secretes an extracellular matrix rich in collagen and other extracellular matrix macromolecules. Migrates and proliferates readily in wounded tissue and in tissue culture. Fibronectin, extracellular matrix protein that binds to integrants on cell surfaces, helping cells to adhere to the matrix. Fibrous protein, a protein with an elongated shape, typically one such as collagen or intermediate filament protein that is able to associate into long filamentous structures. Filipodium, plural Filipodia, long, thin actin containing extensions on the surface of an animal cell. Sometimes has an exploratory function, as in a growth cone. Flagellum, plural flagella, long whip-like protrusion that drives a cell through a fluid medium by its speeding. Eukaryotic flagella are longer versions of cilia. Bacterial flagella are completely different, being smaller and simpler in construction. Free energy, G. Energy that can be extracted from a system to do useful work, such as driving a chemical reaction. The standard free energy of a substance, G degrees, is its free energy measured at a defined concentration, temperature, and pressure. Free energy change, delta G. Delta G, the difference in free energy between reactant and product molecules in a chemical reaction. A large negative value of delta G indicates that the reaction has a strong tendency to occur. The standard free energy change, delta G degrees, is the free energy change measured at defined concentration, temperature, and pressure. G delta G degrees. See free energy change. G protein coupled receptor. Cell surface receptor that associates with an intracellular trimeric GTP binding protein, G protein, after receptor activation by an extracellular ligand. These receptors are 7-pass transmembrane proteins. GAG, see glycosaminoglycan. Gain of function mutation, a mutation that increases the activity of a gene or makes it active in inappropriate circumstances. Such mutations are usually dominant. Gamete, see germ cell. Gap junction, communication cell-cell junction that allows ions and small molecules to pass from the cytoplasm of one cell to the cytoplasm of the next. GDP, guanosine 5' diphosphate, nucleotide that is produced by the hydrolysis of the terminal phosphate of GTP, a reaction that also produces inorganic phosphate. When free in solution, GDP is rapidly rephosphorylated to GTP, usually by the transfer of the terminal phosphate from ATP in the reaction ATP plus GDP to ADP plus GTP. Gene, region of DNA that controls a discrete hereditary characteristic of an organism, usually responsible for specifying a single protein or RNA molecule. Gene duplication, the accidental duplication of a gene or of a stretch of DNA containing several genes in the genome. The resulting 2 copies of the gene can then diverge by the accumulation of mutations. Gene families arise through a series of gene duplication events. Gene expression, the process by which a gene makes its effect on a cell or organism by directing the synthesis of a protein or an RNA molecule with a characteristic activity. Gene replacement, the replacement of a normal gene in an organism with one that has been mutated in vitro, used to investigate the gene's function. General transcription factors, proteins that assemble on the promoters of many eukaryotic genes near the start site of transcription and load the RNA polymerase in the correct position. Genetic code, set of rules specifying the correspondence between nucleotide triplets, codons in DNA or RNA, and amino acids in proteins. Genetic engineering, see recombinant DNA technology. Genetic instability, increased mutation rate seen, for example, in cancer cells, resulting from the presence of mutations that interfere with the accurate replication and maintenance of the genome. Genetic map, a graphic representation of the order of genes in chromosomes, spaced according to the amount of recombination that occurs between them. Genetic screen, a search through a collection of mutants for a particular phenotype. Genetics, the study of the genes of an organism based on heredity and variation. Genome, the total genetic information carried by a cell or an organism, or the DNA molecules that carry this information. Genotype, set of genes carried by an individual cell or organism. Germ cell, gamete, cell type in a diploid organism that carries only one set of chromosomes and is specialized for sexual reproduction, a sperm or an egg. Germline, the lineage of reproductive cells that contributes to the formation of a new generation of organisms, as distinct from somatic cells, which form the body and leave no descendants in the next generation. Globular protein. Any protein with an approximately rounded shape. Most enzymes are globular. Gluconeogenesis. The synthesis of glucose from small organic molecules, such as lactate, pyruvate, or amino acids. Glucose. 6-carbon sugar that plays a major role in the metabolism of living cells, stored in polymeric form as glycogen in animal cells and as starch in plant cells. See panel 2.3, pages 68 to 69. Glycogen. Polysaccharide composed exclusively of glucose units, used to store energy in animal cells. Large granules of glycogen are especially abundant in liver and muscle cells. Glycolipid. Membrane lipid molecule with a short carbohydrate chain attached to a hydrophobic tail. Glycolysis. Ubiquitous metabolic pathway in the cytosol in which sugars are incompletely degraded with production of ATP. Literally, sugar splitting. Glycoprotein. Any protein with one or more covalently linked oligosaccharide chains. Includes most secreted proteins and most proteins exposed on the outer surface of the plasma membrane. Glycosaminoglycan, GAG. Family of high molecular weight polysaccharides containing amino sugars, found as protective coats around animal cells. Golgi apparatus. Membrane-enclosed organelle in eukaryotic cells where the proteins and lipids made in the endoplasmic reticulum are modified and sorted for transport to other sites, named after its discoverer, Camillo Golgi. G1 phase. Gap 1 phase of the eukaryotic cell cycle between the end of cytokinesis and the start of DNA synthesis. G1-CDK. Cyclin-dependent kinase whose activity drives the cell through G1 phase. G1-SCDK. Cyclin-dependent kinase whose activity triggers entry into S phase of the cell cycle. G2 phase. Gap 2 phase of the eukaryotic cell cycle, between the end of DNA synthesis and the beginning of mitosis. G protein. One of a large family of GTP-binding proteins composed of 3 different subunits, heterotrimeric GTP-binding proteins that are important intermediaries in intracellular signaling pathways, usually activated by the binding of a hormone or other ligand to a transmembrane receptor. Green fluorescent protein, GFP, fluorescent protein from a jellyfish that is widely used as a marker for monitoring the movement of proteins in living cells. Group, see chemical group. Growth factor, extracellular polypeptide signaling molecule that stimulates a cell to grow or proliferate. Examples are epidermal growth factor, EGF, and platelet-derived growth factor, PDGF. GTP, guanosine 5' triphosphate, major nucleoside triphosphate used in the synthesis of RNA and in some energy transfer reactions. Also has a special role in microtubule assembly, protein synthesis, and cell signaling. GTP binding protein, an allosteric protein whose conformation and activity are determined by its association with either GTP or GDP, includes many proteins involved in cell signaling, such as ROS and G proteins. Haploid, a cell or organism with only one set of chromosomes, as in a sperm cell or a bacterium. See also diploid. Haplotype block, a combination of alleles and other DNA markers on a chromosome that has been inherited in a large linked block, undisturbed by genetic recombination, across many generations. Helix, an elongated structure in which a filament or thread twists in a regular fashion around a central axis. Alpha helix, see alpha helix. Hemidesmosome, specialized anchoring cell junction between an epithelial cell and the underlying basal lamina. Heredity, the transmission from one generation to another of genetic factors that determine individual characteristics, responsible for the similarity between parents and children. Heterochromatin, region of a chromosome that remains unusually condensed and transcriptionally inactive during interphase. Heterozygous, describes an organism with dissimilar alleles for a given gene. High-energy bond, covalent bond whose hydrolysis releases an unusual large amount of free energy under the conditions existing in a cell. Examples include the phosphodiester bonds in ATP and the thioester linkage in acetyl-CoA. Histone, one of a group of abundant basic proteins rich in arginine and lysine that are associated with DNA and chromosomes to form nucleosomes. Histone deacetylase, enzyme that removes acetyl groups from lysines present in histones. The acetylation state of histones acts as a signal that attracts other proteins that activate or repress transcription. Homologue. 1. See homologous chromosome. 2. Any structure or macromolecule that has a close similarity to another as a result of common ancestry. Homologous. Describes organs or molecules that are similar because of their common evolutionary origin. Specifically, it describes similarities between protein sequences or nucleic acid sequences. Homologous chromosome. One of the two copies of a particular chromosome in a diploid cell, one from the father and the other from the mother. Homologous gene. See homologous. Homologous recombination. Genetic exchange between a pair of identical or very similar DNA sequences, typically located on a pair of homologous chromosomes. A similar process is used to repair double-strand breaks in DNA. Homozygous. Describes an organism having identical alleles for a given gene. Horizontal gene transfer. Process through which DNA is passed from one organism to another, permanently changing the DNA composition of the recipient. This contrasts with vertical gene transfer, which refers to the inheritance of genes from parent to progeny. Hormone. A chemical substance produced by one set of cells in a multicellular organism and transported via body fluids to target tissues on which it exerts a specific effect. Hybridization. Experimental process in which two complementary nucleic acid strands form a double helix, a powerful technique for detecting specific nucleotide sequences. Hydrogen bond. A weak, non-covalent chemical bond between an electronegative atom, such as nitrogen or oxygen, and a hydrogen atom bound to another electronegative atom. Hydrogen ion. Commonly used term for a proton, H+, in aqueous solution, the basis of acidity. Since a proton readily combines with a water molecule to form H3O+, it is more accurate to call it a hydronium ion. See panel 2, pages 66 to 67. Hydrolysis. Adjective, hydrolytic. Cleavage of a covalent bond with accompanying addition of water. H being added to one product of the cleavage and OH to the other. Hydronium ion, H3O+. The form taken by a proton H+ in aqueous solution. See panel 2, pages 66 to 67. Hydrophilic. Polar molecule or part of a molecule that forms enough hydrogen bonds to water to dissolve readily in water. Literally, water-loving. Hydrophobic. Non-polar molecule or part of a molecule that cannot form favorable bonding interactions with water molecules and therefore does not dissolve in water. Literally, water-hating. Hydroxyl, OH. Chemical group consisting of a hydrogen atom linked to an oxygen as an alcohol. See panel 2.1, pages 64 to 65. Induced pluripotent stem cell, iPS cell. Somatic cell reprogrammed into an embryonic stem cell-like state by artificially introducing a particular set of genes. Initiation factor. protein that promotes the proper association of ribosomes with mRNA and is required for the initiation of protein synthesis. Initiator tRNA. Special tRNA that initiates translation. It always carries the amino acid methionine. Inositol. Sugar molecule with 6 hydroxyl groups that forms the framework for inositol phospholipids. Inositol 1,4,5-trisphosphate, IP3. Small intracellular signaling molecule produced during activation of the inositol phospholipid signaling pathway. Causes Ca2+ release from the endoplasmic reticulum. Inositol phospholipids, phosphoinositides. Minor lipid components of plasma membranes that contain phosphorylated inositol derivatives, important both for distinguishing between intracellular membranes and for signal transduction in eukaryotic cells. In situ hybridization. Technique in which a single-stranded RNA or DNA probe is used to locate a gene or an mRNA molecule in an entire cell or tissue. Integrin. Family of transmembrane proteins present on cell surfaces that enable cells to adhere to each other and to the extracellular matrix, being also involved in cell signaling. Intermediate filament. Fibrous protein filament, about 10 nanometers in diameter, that forms rope-like networks in animal cells, often used as a structural element that resists tension applied to the cell from the outside. Interphase. Long period of the cell cycle between one mitosis and the next. Includes G1 phase, S phase, and G2 phase. Interphase chromosome. State of a eukaryotic chromosome when the cell is between divisions. These chromosomes are active in transcription and much more extended than mitotic chromosomes. Intron. Non-coding region of a eukaryotic gene that is transcribed into an RNA molecule but is then excised by RNA splicing to produce mRNA. Intracellular signaling molecule. Molecule, usually a protein, that is part of the mechanism for transducing and transmitting signals inside a cell. Intracellular signaling pathway. The set of proteins and small molecule second messengers that interact with each other to relay a signal from the cell membrane to its final destination in the cytoplasm or nucleus. In vitro. Term used by biochemists to describe a process taking place in an isolated, cell-free extract. Also used by cell biologists to refer to cells growing in culture, in vitro, as opposed to in an organism, in vivo, Latin for in glass. In vivo, in an intact cell organism, Latin for in life. Ion, an atom carrying an electric charge, either positive or negative. Ion channel, transmembrane protein or protein complex that forms a water-filled channel across the lipid bilayer through which specific inorganic ions can diffuse down their electrochemical gradients. Ion channel coupled receptor, transmembrane receptor protein or protein complex that forms a gated ion channel that opens in response to the binding of a ligand to the external face of the channel. Ionic bond, attractive force that holds together two ions, one positive, the other negative. IP3, C-inositol 1,4,5-trisphosphate. Iron-sulfur center, one of a family of electron transporters containing iron atoms linked to sulfur atoms and cysteine side chains, found in electron transport chains such as those in mitochondria and chloroplasts. Isomer, stereoisomer, one of two or more substances that contain the same atoms and have the same molecular formula, such as C6H12O6, but differ in the spatial arrangement of these atoms. Optical isomers differ only by being mirror images of each other. Isotopes, two or more forms of an atom that have the same chemistry but differ in atomic weight, may be either stable or radioactive. K, see equilibrium constant. K+, potassium ion, a major ionic constituent of living cells. K subscript M, the concentration of substrate at which an enzyme works at half its maximum rate. Large values of K subscript M usually indicate that the enzyme binds to its substrate with relatively low affinity. Karyotype, a display of the full set of chromosomes of a cell arranged with respect to size, shape, and number. Kilocalorie, kcal, unit of heat equal to 1,000 calories, often used to express the energy content of food or molecules. Bond strengths, for example, are measured in kilocalories per mole. An alternative unit in wide use is the kilojoule. Kilojoule, standard unit of energy equal to 0.239 kilocalories. Kinase, see protein kinase. Kinesin, a large family of motor proteins that use the energy of ATP hydrolysis to move along a microtubule. Kinetochore, complex protein containing structure on a mitotic chromosome to which microtubules attach. The kinetochore forms on the part of the chromosome known as the centromere. Knockout mouse, a genetically engineered mouse in which a specific gene has been inactivated, for example by introducing a deletion in its DNA. Lagging strand, one of the two newly made strands of DNA found at a replication fork. The lagging strand is made in discontinuous lengths that are later joined covalently. Lamellipodium, dynamic sheet-like extension on the surface of an animal cell, especially one migrating over a surface. Law of independent assortment, the second law of heredity, derived by Mendel, which states that during gamete formation the alleles for different traits segregate independently of one another. Law of segregation, the first law of heredity derived by Mendel, which states that the maternal and paternal alleles for a trait separate from one another during gamete formation and then reunite during fertilization. Leading strand, one of the two newly made strands of DNA found at a replication fork. The leading strand is made by continuous synthesis in the 5' to 3' direction. Ligand, general term for a molecule that binds to a specific site on a protein. Ligand-gated channel, an ion channel that opens when it binds a small molecule such as neurotransmitter. Ligase, enzyme that joins two DNA strands together end to end. Lipid, organic molecule that is insoluble in water but dissolves readily in nonpolar organic solvents. One class, the phospholipids, forms the structural basis of biological membranes. Lipid bilayer, thin bimolecular sheet of mainly phospholipid molecules that forms the structural basis for all cell membranes. The two layers of lipid molecules are packed with their hydrophobic tails pointing inward and their hydrophilic heads outward, exposed to water. Local mediator, secreted signal molecule that acts as a short range on adjacent cells. Loss of function mutation, a mutation that reduces or eliminates the activity of a gene. Such mutations are usually recessive. The organism can function normally as long as it retains at least one normal copy of the affected gene. Lumen, cavity enclosed by an epithelial sheet in a tissue or by a membrane in a cell, as in the lumen of an endoplasmic reticulum. From Latin, lumen, light or opening. Lymphocyte, white blood cell that mediates the immune response to a foreign molecule, an antigen. Lymphocytes are either of the antibody-secreting B cell type or the T cell type that regulates responses and also forms a cell-mediated immune response system. Lysosome, intracellular membrane-enclosed organelle containing digestive enzymes, typically those most active at the acid pH found in these organelles. MCDK, active protein complex formed at the start of M phase of the cell cycle by an M cyclin and the mitotic cyclin-dependent protein kinase, CDK. M cyclin, cyclin protein that binds to mitotic CDK to form MCDK at the start of M phase of the cell cycle. M phase, period of the eukaryotic cell cycle during which the nucleus and cytoplasm divide. Macromolecule, Molecules such as a protein, nucleic acid, or polysaccharide with a molecular mass greater than a few thousand daltons. From Greek, makros, large. Macrophage. Cell found in animal tissues that is specialized for the uptake of particular material by phagocytosis, derived from a type of white blood cell. MAP kinase. Mitogen-activated protein kinase. Protein kinase that performs a crucial step in relaying signals from cell surface receptors to the nucleus. It is the final kinase in a 3-kinase sequence called the MAP kinase cascade. Matrix. Most generally, a space within which something is formed. In cell biology, this word often refers to the large internal compartment of the mitochondrion. The mitochondrial matrix contains a concentrated mixture of special enzymes that catalyze oxidation reactions, as well as the mitochondrial genome and the proteins needed to express mitochondrial genes. See Figure 14.4. Meiosis. Special type of cell division by which eggs and sperm cells are made. 2 successive nuclear divisions with only 1 round of DNA replication generates 4 haploid daughter cells from an initial diploid cell. From Greek, meiosis, diminution. Membrane. Thin sheet of lipid molecules and associated proteins that enclose all cells and form the boundaries of many eukaryotic organelles. Membrane-enclosed organelle. Any organelle in the eukaryotic cell that is surrounded by a lipid bilayer membrane. For example, the endoplasmic reticulum, Golgi apparatus, and lysosome. Membrane domain. Functionally specialized region in a cell membrane characterized by the presence of particular proteins. Membrane potential. Voltage difference across a membrane due to a slight excess of positive ions on one side and of negative ions on the other. A typical membrane potential for an animal cell plasma membrane is negative 60 millivolts, inside negative, measured relative to the surrounding fluid. Membrane protein. A protein associated with a lipid bilayer, can be either integral, transmembrane, or peripheral. Membrane transport protein. Any protein embedded in a membrane that serves as a carrier of ions or small molecules from one side to the other. Messenger RNA, mRNA. RNA molecule that specifies the amino acid sequence of a protein, produced by RNA splicing in eukaryotes, from a larger RNA molecule made by RNA polymerase as a complementary copy of DNA. It is translated into protein in a process catalyzed by ribosomes. Metabolic pathway. Sequence of enzymatic reactions in which the product of one reaction is the substrate of the next. Metabolism. The sum total of the chemical reactions that take place in the cells of a living organism resulting in growth, division, energy production, excretion of waste, and so on. Metaphase. Stage of mitosis at which chromosomes are firmly attached to the mitotic spindle at its equator but have not yet segregated toward opposite poles. Metastasis. The spreading of cancer cells through the body from the initial site of the tumor. Methyl CH3 group. Hydrophobic chemical group derived from methane, CH4. See panel 2.1, pages 64 to 65. Micro. Prefix denoting 10 to the negative 6. Micrograph. Picture taken through a microscope, either a light micrograph or an electron micrograph, depending upon the type of microscope used. Micrometer. Unit of measurement often applied to cells and organelles, equal to 10 to the negative 6 meter or 10 to the negative 4 centimeter. MicroRNA, miRNA. Small non-coding RNAs that control gene expression by base pairing with specific mRNAs to regulate their stability and their translation. Microscope. Instrument for viewing extremely small objects. A light microscope utilizes a focused beam of visible light and is used to examine cells and organelles. An electron microscope utilizes a beam of electrons and can be used to examine objects as small as individual molecules. Microtubule. Long, stiff, cylindrical structure composed of the protein tubulin, used by eukaryotic cells to regulate their shape and control their movements. Milli, prefix denoting 10 to the negative 3. Mismatch repair, important error correction mechanism in DNA replication that is triggered by the mismatch of non-complementary base pairs. Mitochondrion, plural mitochondria, membrane-enclosed organelle about the size of a bacterium that carries out oxidative phosphorylation and produces most of the ATP in eukaryotic cells. Mitogen, an extracellular signal molecule that stimulates cell proliferation. Mitosis, division of the nucleus of a eukaryotic cell, which involves condensation of the DNA into visible chromosomes. From Greek mitos, a thread, referring to the thread-like appearance of the condensed chromosomes. Mitotic chromosome, highly condensed duplicated chromosome with the two new chromosomes, also called sister chromatids, still together at the centromere, a chromosome during one of the stages of mitosis. Mitotic spindle, array of microtubules that associated molecules that forms between the opposite poles of a eukaryotic cell during mitosis. During the separation of the duplicated chromosomes, the spindle serves to move the two centromeres, to move the two chromosome sets apart. Mobile genetic element, short segment of DNA that can move, sometimes through an RNA intermediate, from one location in a genome to another. They are an important source of genetic variation in most genomes. Model organism, an organism selected for intensive study as a representative of a large group of species. Examples are the mouse, representing mammals, the yeast, Saccharomyces cerevisiae, representing a unicellular eukaryote, and Escherichia coli, representing bacteria. Mole, molar grams of a substance, where molar is its relative molecular mass, molecular weight. This will be 6 times 10 to the 23 molecules of the substance. Molecular switch, protein or protein complex that operates in an intracellular signaling pathway and can reversibly switch between an active and an inactive state. Molecular weight, mass of a molecule expressed in daltons. Molecule, group of atoms joined together by covalent bonds. Monomer, small molecule that can be linked to others of a similar type to form a large molecule. Polymer. Monomeric GTPase, small single subunit GTP binding protein. Proteins of this family, such as ROS and Rho, are part of the many different signaling pathways. Motor protein, protein such as myosin or kinesin that uses energy derived from ATP hydrolysis to propel itself along a protein filament or polymeric molecule. mRNA, see messenger RNA. Mutation, a randomly produced heritable change in the nucleotide sequence of a chromosome. Myofibril, long, highly organized bundle of actin, myosin, and other proteins in the cytoplasm of muscle cells that contracts by a sliding filament mechanism. Myosin, type of motor protein that uses ATP to drive movements along actin filaments. Myosin II is a large protein that forms the thick filaments of skeletal muscle. Smaller myosins, such as myosin I, are widely distributed and responsible for many actin-based movements. N-terminus, amino terminus, the end of a polypeptide chain that carries a free alpha amino group. Na+, sodium ion, a major ionic constituent of living cells. NAD+, nicotine adenine dinucleotide, activated carrier molecule that participates in an oxidation reaction by accepting a hydride ion, H-, from a donor molecule, thereby producing NADH, widely used in the energy-producing breakdown of sugar molecules. See Figure 334. NADPH, nicotine adenine dinucleotide phosphate, a carrier molecule closely related to NADH used as an electron donor in biosynthetic pathways In the process, it is oxidized to NADP+. See Figure 335. Na plus K plus pump. Na plus K plus ATPase. Sodium pump. Transmembrane carrier protein, found in the plasma membrane of most animal cells, that pumps Na plus out of the K and K plus into the cell. That pumps Na plus out of and K plus into the cell, using the energy derived from ATP hydrolysis. Nanometer. Unit of length commonly used to measure molecules in cell organelles. 1 nanometer equals 10 to the negative 3 micrometers, equals 10 to the negative 9 meters. Nernst equation. Quantitative expression that relates the equilibrium ratio of concentrations of an ion on either side of a permeable membrane to the voltage difference across the membrane. Nerve cell. See neuron. Nerve terminal. The ending of an axon from which signals are sent to adjoining cells, usually at a synapse. Neuron. Nerve cell. Cell with long processes specialized to receive, conduct, and transmit signals in the nervous system. Neurotransmitter. Small signaling molecules secreted by a nerve cell at a chemical synapse to signal to the postsynaptic cell. Examples include acetylcholine, glutamate, GABA, and glycine. Nitric oxide, NO. Small, highly diffusible molecule widely used as an intracellular signal. Nitrogen fixation. Conversion of nitrogen from the atmosphere into nitrogen-containing organic molecules by soil bacteria and cyanobacteria. NO. See nitric oxide. Non-covalent bond. Chemical bond in which, in contrast with a covalent bond, no electrons are shared. Non-covalent bonds are relatively weak, but they can sum together to produce strong, highly specific interactions between molecules. Examples are hydrogen bonds and van der Waals interactions. Non-homologous end joining. Mechanism for repairing double-strand breaks in DNA in which the two broken ends are brought together and rejoined without requiring sequence homology. Nonpolar. Describes a molecule that lacks a local accumulation of positive or negative charge. Nonpolar molecules are generally insoluble in water. Nuclear envelope. Double membrane surrounding the nucleus, consists of outer and inner membranes perforated by nuclear pores. Nuclear lamina. Fibrous layer on the inner surface of the inner nuclear membrane formed as a network of intermediate filaments made from nuclear lamins. Nuclear magnetic resonance, NMR spectroscopy. Technique used for determining the three-dimensional structure of a protein. It is performed in solution without requiring a protein crystal. Nuclear pore. Channel through the nuclear envelope that allows selected large molecules to move between the nucleus and the cytoplasm. Nuclear receptor. Receptor proteins present inside a eukaryotic cell that can bind to signal molecules that enter the cell, such as steroid hormones. The complex of nuclear receptor and signal molecules subsequently acts as a transcription regulator. Nucleic acid. RNA or DNA consists of a chain of nucleotides joined together by phosphodiester bonds. Nucleolus. Large structure in the nucleus where ribosomal RNA is transcribed and ribosomal subunits are assembled. Nucleoside. Compound composed of a purine or pyrimidine base linked to either a ribose or a deoxyribose sugar. See panel 2.6, pages 74 to 75. Nucleosome. Bead-like structural unit of a eukaryote chromosome composed of a short length of DNA wrapped around a core of histone proteins, the fundamental subunit of chromatin. Nucleotide. Nucleoside with a series of one or more phosphate groups joined by an ester linkage to the sugar moiety. DNA and RNA are polymers of nucleotides. Nucleus. The major organelle of a eukaryotic cell, which contains DNA organized into chromosomes. Also, when referring to an atom, the central mass built from neutrons and protons. Okazaki fragments. Short length of DNA produced on the lagging strand during DNA replication. Adjacent fragments are rapidly joined together by DNA ligase to form a continuous DNA strand. Oligo. Prefix that denotes a short polymer, oligomer, may be made of amino acids, oligopeptide, sugars, oligosaccharide, or nucleotides, oligonucleotide, from Greek oligos, few or little. Oncogene. Any abnormally activated gene that can make a cell cancerous, typically a mutant from a normal gene, proto-oncogene, involved in the control of cell growth or division. Organic chemistry. The branch of chemistry concerned with compounds made of carbon, includes essentially all of the molecules from which living cells are made, apart from water and metal ions such as Na+. Organelle. A discrete structure or subcompartment of a eukaryotic cell, especially one that is visible in the light microscope, that is specialized to carry out a particular function. Examples include mitochondria and the Golgi apparatus. Origin recognition complex, ORC. Large protein complex that is bound to the DNA at origins of replication in eukaryotic chromosomes throughout the cell cycle. Osmosis. Net movement of water molecules across a semi-permeable membrane driven by a difference in concentration of solute on either side. The membrane must be permeable to water, but not to the solute molecules. Osmotic pressure. Pressure that must be exerted on the low solute concentration side of a semi-permeable membrane to prevent the flow of water across the membrane as a result of osmosis. Oxidation. Loss of electron density from an atom, as occurs during the addition of oxygen to a carbon atom or when a hydrogen is removed from a carbon atom, the opposite of reduction. See Figure 311. Oxidative phosphorylation. Process in bacteria and mitochondria in which ADP formation is driven by the transfer of electrons from food molecules to molecular oxygen. Involves the intermediate generation of a pH gradient across a membrane and chemiosmotic coupling. P53, regulatory protein that responds to the presence of DNA damage, preventing the cell from entering S phase until the damage has been repaired. Pairing. In a genetic sense, the event early in meiosis in which two homologous chromosomes line up together to form a duplicated structure. See also base pair. Passive transport. The movement of a small molecule or ion across a membrane due to a difference in concentration or electrical charge. Patch clamp recording. Technique in which the tip of a small glass electrode is sealed onto a patch of cell membrane, thereby making it possible to record the flow of current through individual ion channels in the patch. PCR. See polymerase chain reaction. Pedigree. The line of descent or ancestry of an individual animal. Peptide bond. Chemical bond between the carbonyl group of one amino acid and the amino group of a second amino acid. A special form of amide linkage. See panel 2.5, pages 72 to 73. Peroxisome. Small membrane enclosed organelle that uses molecular oxygen to oxidize organic molecules. Contains some enzymes that produce hydrogen peroxide, H2O2, and others that degrade it. Phagocytic cell. A cell such as a macrophage or neutrophil that is specialized to take up particles in microorganisms by phagocytosis. Phagocytosis. The process by which particulate material is engulfed, eaten by a cell. Prominent in predatory cells such as amoeba proteus and in cells of the vertebrate immune system such as macrophages. Phenotype. The observable character of a cell or organism. Phosphatidylcholine. Common phospholipid present in abundance in most biological membranes. See Figure 11.6. Phosphodiester bond. A covalent chemical bond in which two carbon atoms are held in ester linkage via oxygen atoms to the same phosphate group. Phosphodiester bonds join the adjacent nucleotides in RNA or DNA. See Figure 225. Phosphoinositide 3 kinase, PI3 kinase, enzyme that phosphorylates inositol phospholipids in the plasma membrane in response to signals received by a cell. The phosphorylated lipids become docking sites for intracellular signaling proteins. Phospholipase C, enzyme associated with the plasma membrane that performs a crucial step in inositol phospholipid signaling pathways. Phospholipid, type of lipid molecule used to make biological membranes, generally composed of two fatty acids linked through glyceryl phosphate to one of a variety of polar groups. Phosphorylation, see protein phosphorylation. Photosynthesis, the process by which plants and some bacteria use the energy of sunlight to drive the synthesis of organic molecules from carbon dioxide and water. Photosystem, a large multi-protein complex containing chlorophyll that captures light energy. Phragmoplast, structure made of microtubules and membrane vesicles that forms in the equatorial region of a dividing plant cell and from which the membrane that divides the daughter cells will be made. pH scale, scale used to measure the acidity of a solution. P refers to power of 10, H to hydrogen, defined as a negative logarithm of the hydrogen ion concentration in moles per liter. Thus, an acidic solution with pH 3 will contain 10 to the negative 3 molar hydrogen ions. Phylogenetic tree, chart or family tree showing the evolutionary history of a group of organisms. Pinocytosis, type of endocytosis in which soluble materials are taken up from the environment and incorporated into vesicles for digestion. Literally, cell drinking. pKa, see protein kinase C. Plasma membrane, the membrane that surrounds a living cell. Plasmid, small circular DNA molecule that replicates independently of the genome, used extensively as a vector for DNA cloning. Plasmodesma, plural, plasmodesmata, cell-cell junction in plants in which a channel of cytoplasm lined by membrane connects two adjacent cells through a small pore in their cell walls. Point mutation, change in a single nucleotide pair in a DNA sequence. Polar, describes a molecule or a covalent bond in a molecule in which bonding electrons are attracted more strongly to specific atoms, thereby creating an uneven or polarized distribution of electric charge. Polarity refers to a structure such as an actin filament or a fertilized egg that has an inherent asymmetry, so that one end can be distinguished from the other. Polymer, large and usually linear molecule made by the repetitive assembly using covalent bonds of multiple identical or similar subunits, monomers. Polymerase, general term for an enzyme that catalyzes addition of subunits to a polymer. DNA polymerase, for example, makes DNA, and RNA polymerase makes RNA. Polymerase chain reaction, PCR, technique for amplifying specific regions of DNA by multiple cycles of DNA synthesis, each followed by a brief heat treatment to separate complementary DNA strands. Polymorphisms, the case where two or more variants, alleles of a gene or variants of a DNA sequence coexist in the population and are each relatively common. Polynucleotide, a molecular chain of nucleotides chemically bonded by a series of phosphodiester linkages, a strand of RNA or DNA. Polypeptide, polypeptide chain, linear polymer composed of multiple amino acids. Proteins are composed of one or more long polypeptide chains. Polypeptide backbone, the chain of atoms containing repeating peptide bonds that runs through a protein molecule and to which the amino acid side chains are attached. Polysaccharide, linear or branched polymer composed of sugars. Examples are glycogen, hyaluronic acid, and cellulose. Polysome, polyribosome, messenger RNA molecule with multiple attached ribosomes that is engaged in protein synthesis. Positive feedback loop, situation in which the end product of a reaction stimulates its own production. Post-transcriptional control, regulation of gene expression that occurs after transcription of the gene has begun. Examples are regulation of RNA splicing and other RNA processing events, and the regulation of translation by microRNA. Primary transcript, see transcription. Primer, in DNA replication, a short length of RNA made at the beginning of a DNA synthesis event catalyzed by DNA polymerase. These RNA primers are subsequently removed and filled in with DNA. Prokaryote, major category of living cells distinguished by the absence of a nucleus. Prokaryotes comprise the archaea and the eubacteria, commonly called bacteria, two of the three domains of life. Processive, describes a protein that performs repeated rounds of catalysis or conformational changes while still attached to a polymer, a characteristic of motor proteins involved in transport, such as kinesin. Programmed cell death, see apoptosis. Prometaphase, stage of mitosis that precedes metaphase. Promoter, nucleotide sequence in DNA to which RNA polymerase binds to begin transcription. Proofreading, the process by which DNA polymerase corrects its own errors as it moves along DNA. Prophase, first stage of mitosis during which the chromosomes are condensed but not yet attached to a mitotic spindle, also a superficially similar stage in meiosis. Protease, proteinase, proteolytic enzyme, enzymes such as trypsin that degrades proteins by hydrolyzing some of their peptide bonds. Proteasome, large protein complex in the cytosol that is responsible for degrading cytosolic proteins that have been marked for destruction by ubiquitination or by some other means. Protein, the major macromolecular constituent of cells. Each protein is composed of one or more linear chains of amino acids linked together by peptide bonds in a specific sequence. The amino acid chain is folded into a three-dimensional shape that is unique to the given protein and determines its function. Protein domain, see domain. Protein complex, see complex. Protein family, a group of proteins in an organism with a similar amino acid sequence. The similarity is thought to reflect the evolution of the genes that encode the proteins from a common ancestor gene through a process of gene duplication followed by gene divergence. Usually, the different members of a protein family will have related but distinct functions. For example, each member of the protein kinase family carries out a similar phosphorylation reaction, but the substrates and regulation differ for each enzyme. Protein kinase, one of a very large number of enzymes that transfers the terminal phosphate group of ATP to a specific amino acid side chain on a target protein. Protein kinase A, see cyclic AMP-dependent protein kinase. Protein kinase C, PKC, enzyme that phosphorylates target proteins in response to a rise in diacylglycerol and Ca2+ ions. Protein machine, a set of protein molecules that bind to each other in specific ways, so that concerted movements within the protein complex can carry out a sequence of reactions with unusual speed and effectiveness. A large number of the central reactions of the cell are catalyzed by such protein machines, with protein synthesis and DNA replication being particularly well understood examples. Protein phosphatase, phosphoprotein phosphatase, enzyme that removes, by hydrolysis, a phosphate group from a protein, often with high specificity for the phosphorylated site. Protein phosphorylation, the covalent addition of a phosphate group to a side chain of a protein catalyzed by a protein kinase. Phosphorylation usually alters the activity or properties of the protein in some way. Proteoglycan, molecule consisting of one or more glycosaminoglycan chains attached to a core protein. Proteolysis, degradation of a protein by means of a protease. Proteomics, the large-scale study of proteins, investigating many different proteins in a cell or tissue simultaneously. Proton, subatomic particle found in the atomic nucleus. Also exists as an independent chemical species as a positive hydrogen ion, H+. Proto-oncogene, see oncogene. Protozoan, a member of the protozoa, free-living, non-photosynthetic, single-celled, motile eukaryotic organisms. Many protozoans, such as paramecium or amoeba, live by feeding on other organisms. Purifying selection, process of selection during evolution in which individuals carrying mutations that interfere with important functions are eliminated. Purine, one of the two categories of nitrogen-containing ring compounds found in DNA and RNA. Examples are adenine and guanine. See panel 2.6, pages 74 to 75. Pyrimidine, one of the two categories of nitrogen-containing ring compounds found in DNA and RNA. An example is cytosine. See panel 2.6, pages 74 to 75. Pyruvate, metabolite formed from the breakdown of glucose that provides a crucial link to the citric acid cycle and many biosynthetic pathways. Quinone, small, lipid-soluble, mobile electron carrier molecule found in the respiratory and photosynthetic electron transport chains. See figure 1420. Rab protein, a family of small GTP-binding proteins present on the surfaces of transport vesicles and organelles that serve as molecular markers identifying each membrane type. Rab proteins help to ensure that membrane vesicles fuse only with the correct membrane. ROS, one of a large family of small GTP-binding proteins, also called the monomeric GTPases, that help relay signals from cell surface receptors to the nucleus. Named for the ROS gene, first identified in viruses that cause rat sarcomas. Reaction center. In photosynthetic membranes, a protein complex that contains a specialized pair of chlorophyll molecules that performs photochemical reactions to convert the energy of photons, light, into high-energy electrons for transport down the photosynthetic electron transport chain. Reading frame. The set of successive triplets in which a string of nucleotides is translated into protein. An mRNA molecule is read in one of three possible reading frames, depending on the starting point. Receptor. A cell, such as the photoreceptor cells of the eye, or cellular component, such as a receptor protein, that detects an external signal and triggers a specific cellular response. Receptor-mediated endocytosis. Mechanism of selective uptake of material by animal cells in which a macromolecule binds to a receptor in the plasma membrane and enters the cell in a clathrin-coated vesicle. Receptor protein. Protein that detects a stimulus, usually a change in concentration of a specific molecule, and then initiates a response in the cell. Cell surface receptors, such as the acetylcholine receptor and the insulin receptor, are located in the plasma membrane, with their ligand-binding site exposed to the external medium. Intracellular receptors, such as steroid hormone receptors, bind ligands that diffuse into the cell across the plasma membrane. Receptor serine threonine kinase. Enzyme-coupled receptor with an extracellular signal binding domain and an intracellular kinase domain that phosphorylates signaling proteins on serine or threonine. Receptor tyrosine kinase, RTK. Enzyme-coupled receptor in which the intracellular domain has a tyrosine kinase activity, which is activated by a ligand binding to the receptor's extracellular domain. Recognition. See molecular recognition. Recombinant DNA. A DNA molecule that is composed of DNA from different sources. Recombinant DNA technology. Genetic engineering. The collection of techniques by which DNA segments from different sources are combined to make new DNA. Recombinant DNAs are widely used in the cloning of genes, in the genetic modification of organisms, and in molecular biology generally. Recombination. Process in which an exchange of genetic information occurs between two chromosomes or DNA molecules. Enzyme-mediated recombination can occur naturally in living cells, or in a test tube using purified DNA and enzymes that break and religate DNA strands. Redox pair. Pair of molecules in which one acts as an electron donor and one as an electron acceptor in an oxidation-reduction reaction. For example, NADH, electron donor, and NAD+, electron acceptor. Redox potential. A measure of the tendency of a given redox pair to donate electrons, act as a reducing agent, or to accept electrons, act as an oxidizing agent. Redox reaction. A reaction in which electrons are transferred from one chemical species to another. An oxidation-reduction reaction. Reduction. Addition of electron density to an atom, as occurs during the addition of hydrogen to a carbon atom or the removal of oxygen from it, the opposite of oxidation. See Figure 311. Regulatory DNA sequence. DNA sequence to which a transcription regulator binds to determine when, where, and in what quantities a gene is to be transcribed into RNA. Regulatory protein code. The set of covalent modifications that a protein has at any given time, which controls the behavior of the protein inside the cell. Replication fork. Y-shaped region of a replicating DNA molecule at which the two daughter strands are formed and separate. Replication origin. Site on a chromosome at which DNA replication begins. Reporter gene. Introduced gene encoding a protein whose activity is easy to monitor experimentally. It is usually joined to a regulatory sequence, which will then switch on the reporter gene in the normal context in which its own gene is usually expressed. Repressor. A protein that binds to a specific regulatory region of DNA to prevent transcription of an adjacent gene. Reproductive cloning. The artificial production of genetically identical copies of an animal by, for example, the transplantation of somatic cell nucleus into an enucleated fertilized egg. Respiration. General term for any process in a cell in which the uptake of molecular oxygen, O2, is coupled to the production of CO2. Restriction nuclease. Restriction enzyme. Nuclease that can cleave a DNA molecule at any site where a specific short sequence of nucleotides occurs. Different restriction nucleases cut at different sequences, extensively used in recombinant DNA technology. Retrotransposon. Type of mobile genetic element that moves by being first transcribed into an RNA copy that is reconverted to DNA by reverse transcriptase and inserted elsewhere in the chromosomes. Retrovirus. RNA containing virus that replicates in a cell by first making a double-stranded DNA intermediate. This DNA is inserted into the cell's chromosome, where it can be maintained for a long time and is transcribed to produce new viral genomes and mRNAs that encode viral proteins. Reverse transcriptase. Enzyme that makes a double-stranded DNA copy from a single-stranded RNA template molecule, present in retroviruses and as part of the transposition machinery of retrotransposons. Rho protein family. Family of small GTPases involved in signaling that causes a rearrangement of the actin cytoskeleton. Ribonucleic acid, cRNA. Ribosomal RNA, rRNA. Any one of a number of specific RNA molecules that form part of the structure of a ribosome and participate in the synthesis of proteins, often distinguished by their sedimentation coefficient, such as 28S rRNA or 5S rRNA. Ribosome. Particle composed of ribosomal RNAs and ribosomal proteins that associates with messenger RNA and catalyzes the synthesis of protein. Riboswitch. Short sequences within some RNAs that change their conformation when specifically bound to small molecules such as metabolites, and in this way regulate transcription or translation. Ribozyme, an RNA molecule possessing catalytic properties. RNA, ribonucleic acid, a usually single-stranded polynucleotide in the form of a chain of covalently linked ribonucleotide subunits. It is synthesized when an RNA polymerase copies the nucleotide sequence of DNA. RNA serves a variety of functions in cells. See, for example, messenger RNA, microRNA, ribosomal RNA, transfer RNA. RNA interference, RNAi. Cellular mechanism activated by double-stranded RNA molecules that results in the destruction of RNAs containing a similar nucleotide sequence. It is widely exploited as an experimental tool for preventing the expression of selected genes. Gene silencing. RNA polymerase. Enzyme that catalyzes the synthesis of an RNA molecule on a DNA template from a nucleoside triphosphate precursors. RNA primer, C primer. RNA processing. Broad term for the modifications from an RNA that an RNA undergoes as it reaches its mature form. For a eukaryotic mRNA, processing typically includes capping, splicing, and polyadenylation. RNA splicing. Process in which intron sequences are excised from RNA molecules in a nucleus during the formation of messenger RNA. Rough endoplasmic reticulum, RER. Region of the endoplasmic reticulum associated with ribosomes and involved in the synthesis of secreted and membrane-bound proteins. rRNA, C ribosomal RNA. RTK, C receptor tyrosine kinase. S phase, period during a eukaryotic cell cycle in which DNA is synthesized. Sarcomere, repeating unit of a myofibril in a muscle cell about 2.5 micrometers long, composed of an array of overlapping thick myosin and thin actin filaments.